Today everyone, let's talk about the process of gene coding.
Gene coding is a process by which the information stored in a gene is translated into functional products such as protein. This process involves multi-steps, starting with transcription of the gene into a messenger RNA molecule by RNA polymerase. During transcription, the DNA double helix unwinds, and the complementary RNA nucleotides are added to the growing mRNA molecule based on the sequence of the DNA template strand. This results in the synthesis of the single-stranded mRNA molecule that contains the genetic information encoded in the gene.
The mRNA molecule is then processed in the nucleus to remove non-coding regions called introns and splice together the coding regions called exons. The processed mRNA is then transported out of the nucleus and into the cytoplasm, where it serves as a template for protein synthesis. Translation is the next step in gene coding and involves the conversion of the mRNA sequence into a specific amino acid sequence that makes up a protein. This process occurs on ribosomes, which read the mRNA triplet codons and assemble the corresponding amino acid into a polypeptide chain. The genetic code, which consists of a specific correspondence between codons and amino acids, ensures that the correct amino acid are added to the growing polypeptide chain.
After translation is complete, the newly synthesized protein may undergo further modification such as folding, glycosylation, or phosphorylation to become functional. So the final protein can be carry out a specific biological function based on its structure or chemical properties. Overall, gene coding is a complex and highly regulated process that ensures the accurate conversion of genetic information into functional protein, essential for the proper functioning of cell and organism.
Hey, it's Professor Dave. Let's talk about DNA transcription and translation.
He knows a lot about the science stuff. Professor Dave explains.
The first step is called transcription. This is the process by which enzymes use one of the strands of DNA within a gene as a template to produce a messenger RNA, or mRNA. To do this, RNA polymerase, with the help of proteins called transcription factors, binds to a specific sequence within the gene, which is called the promoter, and pries the two strands apart. One of the strands will serve as the template strand, or antisense strand, meaning it will be used to generate the mRNA. And the other is the non-template strand, or the sense strand. RNA polymerase doesn't need a primer. It simply initiates mRNA synthesis at the start point and then moves downstream along the gene in a process called elongation, synthesizing the mRNA as it goes, reading the antisense strand from 3' to 5' and generating the mRNA from the 5' end, attaching RNA nucleotides to the 3' end as it goes. This is very similar to the way DNA polymerase synthesizes DNA as it moves along the template strand. The main difference here is that RNA is being synthesized, which, as we recall, will be ribose rather than deoxyribose, and it will have uracil instead of thymine. Unlike replication, RNA polymerase zips DNA back up as it goes, keeping only 10 to 20 bases exposed at a time. Once RNA polymerase reaches the end of the gene, termination occurs. The enzyme detaches from the gene and the DNA is returned to its original state. But we have produced an mRNA. This carries with it the information encoded in the gene, and after a few quick modifications during RNA processing, it will leave the nucleus, where all the genetic material, or chromatin, is, and move into the cytoplasm, where it will find a ribosome. This is where translation occurs. During translation, the mRNA acts as a code for a specific protein. This happens because each set of 3 bases on the mRNA, which we call codons, will code for a specific anticodon, which will be carried by a specific transfer RNA, or tRNA. And each different tRNA is covalently linked to a particular amino acid. The arrangement of the nucleotides into these codons is called the reading frame. Since there are 4 bases and each codon has 3 letters, 4 cubed gives us 64 different possible codons, which is more than enough to code for all the amino acids we need. Here is a table of all the mRNA codons and the amino acids they code for. Notice that there is some redundancy, with multiple codons resulting in the same amino acid. But there is no ambiguity. Each codon corresponds to a particular amino acid. Notice also that some of these codons are special. AUG is the start codon, which initiates translation by coding for methionine. And these 3 are stop codons. These are the ones that terminate translation. Translation will occur inside a ribosome. The small ribosomal subunit binds to an mRNA and an initiator tRNA, which adheres to the start codon. Then the large ribosomal subunit joins to complete the translation initiation complex. Then the tRNA that corresponds to the next codon after the start codon will enter the ribosome. This will carry with it an amino acid, which becomes covalently bound to the methionine from the initiator tRNA. The first tRNA detaches and leaves the ribosome, which has shifted over, making room for the next tRNA. The new amino acid links to the first 2, and this process continues all the way down the mRNA.